hop into the notes. Oh yeah, and a few of you have emailed me since your clicker scores popped up and you asked, why am I missing a couple points here and there? And a lot of those are because people, if you didn't respond to 3/4 of the questions in class on a certain day, then you don't get the participation points. Okay, so I'm always kind of like, all right, I've only got 65 instead of 67. Somebody's not getting their stuff in, so you just got to make sure you get those in. Okay, but there's a total of 40 points in that category in the grade book, and we'll hit, that's pretty much 20 class periods of using your clickers, right? We have around 30 in the semester. Okay, so eventually you'll catch up if you keep using your clickers. So don't stress out about it now, but just use them every day. Answer all the questions, and you will eventually catch up at the end of the semester. Okay. All right. Any questions about any of that? Okay, so we're going to dive into a little bit more on genetics here today. Talk about the cell life cycle, and then talk about some genetic mutations. Okay, so this was what we were just talking about with that S phase, at least, where the DNA replication happens or DNA synthesis happens. Okay, so there's really this whole large process, large time period of cell in the cell cycle called interphase. Right, this is where most of the cell growth is happening, normal functioning, and then the S phase for DNA replication. Okay, but then we've got the mitotic phase, which is where it includes when the cell divides. Okay, so mitosis is really division of the nucleus, whereas cytokinesis, right at the end of the mitotic phase, is the division of the whole cell itself. Okay, and so we're going to kind of go through on the next few slides and just look over some just super basic, super key points of each of the stages of mitosis. Okay. And we'll start with interphase and then get into mitosis. But this is very basic, but I just really want you to know just a couple bullet points from each phase and kind of be able to recognize it in an image as far as what's going on. Okay, so let's take a look at that. Okay, so interphase. We can see the cell nucleus is very solid here, right? The centrioles, which are going to help us with pulling those chromosomes apart, are replicating, so we're getting more of those, getting ready for that. Okay, and this is all preparation for the mitosis phase. Okay, so you can see the cell nucleus there in blue, right? And then those centrioles are developing here, starting to have these strings coming out there. And the key is that they're outside of the nucleus. That's kind of the key part of seeing that there. Okay. All right, so that's interphase. And then we're going to hop on over to prophase. Okay, so prophase, there's kind of early prophase versus late prophase. Okay, so early prophase, the chromosomes have already been duplicated, right? So we've got 2 full sets of chromosomes here. Okay, but now they're going to condense and actually become little worm-looking things, which are which is the way they're going to need to be when we divide the cell. All right. Okay, and then we start to see these very dense parts of the centrioles, these spindles. Okay, and we can see in early prophase, the nuclear membrane is still there, and the nucleus still kind of looks like this round thing. But then in later prophase, the nuclear membrane breaks down, and that's all in preparation for dividing the cell and dividing the nucleus. Okay, so that's prophase. All right, next phase, metaphase. Now the chromosomes are all going to line up at the center of the cell, and these spindle fibers are going to grab on to one of those each, one of those chromatids. In a second, we'll see those spindles are going to retract and pull the chromatids back. Half of them are going to go to one side, half to the other side. Okay, so that's metaphase. The spindle fibers attaching to the chromatids and the chromosomes lining up in the center of the cell. Okay, anaphase. The spindles retract or shorten and pull those chromatids apart. Okay, we can see them starting to be completely separate there. They're dividing, pulling completely apart. Okay, and then that last phase is telophase. And so the new nuclear membranes start to form, and we get cytokinesis. The cell splits into two. Okay, so we can start to see that new nuclear membrane on both sides. Each chromosome, or sorry, each cell now has 23 pairs of chromosomes, or 46 chromosomes altogether. Okay, and then whenever you're ready, you can flip over to the next page with the people on your team and work through those. There's a kind of identifying which one is which, and then you can answer that clicker question there on that slide as well. So for this type of clicker question, you're going to put in all of the letters in the right order and then hit send. Okay. So you're going to press over to the right. That'll take your cursor down, and then you can just scroll through the letters, go to the next one, next one, next one. But you got to hit send at the end of this to make sure it goes in. Oh, dark back here, huh? Let's turn the light on. Notice that, yeah. Yeah, those are those centrioles.
Yeah, so what also these the spindles are like belong, so they don't shorten up or the chromosomes are already they're gymnastics and then okay, so yeah, I agree with you.
Sure, yeah. Yeah, that looks like it. It looks like they're, yeah. So just proteins. Yeah, it has it.
Got that figured out there? Nice. All right, so on your clickers, you're putting in all the letters in order and then hitting send.
Click one of these,
Uh-huh.
and then how would that?
If you want to go past those, then you can just keep scrolling. So you can go all the way to Z, put whatever you want, and then go to the next one over there, put a letter in like that, and just hit send at the end.
Wait.
So once you get all those letters in there, just make sure you hit send.
What are we setting in?
What do you want?
What are we setting in for the?
the order, the correct order of the letters. Yeah.
And what's this right?
You can write me little notes on there if you want, whatever you want.
So we order it from like, let's say A first.
And just go over to the next one. But another one, you can scroll through the whole alphabet here. That way. Yeah. Yeah. Yeah, there you go. You got it. I knew you could figure it out. Then you just hit send when you're ready. Yeah, just like that. And just hit send after you get them all in. You put in the letters in the right order. Right, A is not the first one, right?
Oh, okay.
Yeah. So then go over to the next one. And you can put another letter in, right? You just put them all in the right order. Yeah. All right, put all those letters in and hit send.
I don't know about these. I know about these two for
For what?
I do have 2 points missing on my clicker grade,
Yeah.
but I've done all of the questions.
Probably what I just said, though. There's probably you probably didn't respond to a question on a day where I only had like 1 or 2 questions.
I've definitely, I've definitely responded.
Yeah. I mean, we can look. I'm sure that's what it is, but I wouldn't stress out about it because, like I said, you can miss like 10 classes and still get the full points. So it's not worth the stress. Yeah. All right. So go ahead and hit send. Got 61 of you in there. 63. Yeah, this, and we'll use the clickers in this way at different times, so it's good to figure out how to do it this way. I've had students prank me and write little notes in there. And I put it up on the board and it's something funny, you know. You can write anything. So, not giving you any ideas. But it's harder to put it on the board this way when I'm recording because then we does the infinite screen thing anyway. So, okay, I got 66 of you. Everybody just hit send one more time to make sure we're in there. 67. Anybody else? Raise your hand if you're still working on it. Okay. Okay. You can just hit send. It doesn't really matter too much as long as you get it in there. That's the more important part. Okay, so we've got 54%. There we go, 68. 54% of you saying CFBDAE. Okay. So I'm guessing that's the right answer, but let's take a look. Okay. All right. So first one, C there. All right. So what do we call C? What's that one called? Interphase. Okay. So we got interphase. We can see the nuclear membrane still intact. Centrioles are outside of the nucleus. Okay. Second one was which one? F. Okay. So now we see those, and that one's called, that's early prophase. Okay, so we see those centrioles starting to kind of spread out, kind of multiplying. Okay, so we got that. And then late prophase, which one is that? B, yeah, so up here. Okay, so late prophase is up here. So we see like kind of remnants of the nuclear membrane, but it's been broken down. Okay, the spindles are starting to attach to the chromatids. Okay, and then after that, D, right? D is going to be our next one. So that's going to be our metaphase. Okay, now the chromosomes are completely lined up in the center. Okay, spindles are attached to them, ready to pull back. Okay, next one would be which one? A. So we got A, and that one is our anaphase, right? Okay, so now the spindles are starting to shorten, pulling those chromosomes back. Okay, and then lastly is E, right? So we're starting to get the new nuclear membranes. The cell is starting to divide right there. Okay, so CFBDAE. Okay, just a way for you to work through it and think about these different things with this activity. So any questions on that? Yeah, go on. Yeah, they're just they're lengthening and kind of getting longer. Yeah. I don't know if there's necessarily like, if you count them up, if there's more of them at that point, as far as the images go, as far as them replicating, but they're just kind of, they're definitely longer and lengthening and stretching out. Yeah. So. Yeah. Other questions on that? Okay. All right. So let's talk a little bit more about the cell cycle in regards to these two different types. And so what we're really been talking about is mitosis, okay, which is what most of our cells in our body go through, okay. But then also we have meiosis, okay. Meiosis is happening in our sex cells, our gametes, okay. And so there's a couple of differences in this. We'll get into this more when we talk about the reproductive system next semester, okay. But for now, it's just important to know a couple of the key differences here. Okay, so we definitely get DNA replication happening in both. So in both, we start with 2 full sets of DNA. Okay, but the big difference is meiosis has 2 separate cell divisions, right? There's meiosis 1 and meiosis 2, okay? But there's still only one time that we replicate DNA. So the end result is going to be, first of all, 4 cells in meiosis instead of 2, but then second of all, each of those 4 cells only have half the number of chromosomes that the other two, that the 2 cells, or the cell that they started with. Okay, so these cells here have all 46 chromosomes, but then after we go through the second cell division here, meiosis 2, another DNA replication didn't happen, so we're down to 23 chromosomes in each. Okay, and those are the big differences as far as this image goes that I want you to know. Okay, and then we'll go through a little bit of a compare and contrast here. Okay, so mitosis, the cells that go through this are what known as our somatic cells. Okay, so just like that just means like body cells, right? Cells of most of the cells of the body. Okay, whereas meiosis, it's our germ cells, or also known as gametes, sex cells. Okay, sperm and eggs. Okay, and then we have this term daughter cell, which would be the 2 cells that are there after cell division happened, versus parent cell, which would be the cell that it started with. Okay, daughter cells are genetically identical to the parent cell in mitosis, but in meiosis they're not. Okay, daughter cells' resemblance to each other, okay, genetically identical in mitosis, but in meiosis not. Okay, number of chromosomes in each. So number of chromosomes in daughter versus parent. Okay, they're going to be the same. Both going to be that 46 chromosomes in the somatic cells. In the gametes, it's going to be half. 23 total chromosomes. Okay, number of cells that you end up with 2, known as diploids, which are the ones that have the full set of 46 chromosomes. Okay, and then in meiosis, end up with haploids, which means they kind of have half the number of chromosomes, and there's 4 of them instead of 2. Okay, and the purposes of these for mitosis, it's going to be for really not so much cell growth, but tissue growth. We'll call that. I don't know why I have cell growth there, but tissue growth. Okay, if we're making a tissue bigger, sometimes the way the body does that is by adding more cells to it. Okay, but a lot of times we have to replace cells that are damaged, so that's part of it. So either adding cells to a tissue or replacing damaged or aging cells. Whereas with meiosis, this is the purpose of this is reproduction. And so these germline cells aren't the same thing as the sperm and the egg, but these are the cells that are going to eventually become the sperm and the egg after they go through meiosis 1 and 2. Okay, any questions about those? All right, so just kind of the basic differences between those is what I want you to know. Okay, let's go ahead and do this question here. Should be back to a regular multiple choice question. Go ahead, check it with the people on your team and vote. Just give you about a minute here. No, just one.
Because the G1 and G2.
Yeah, so look back on this one. Interphase is that whole, this whole thing. right? So, interface, yeah.
I take it. It's so weird because I can always keep myself. I was wrong.
All right, go ahead and take your vote when you're ready. Give you about 10 seconds. All right, anybody else? Got 61 of you, 62. Should be a few more. 66, got 2 more still. Anybody else? There we go, 68. I think that's what we had before. Okay, so yeah, 76% of you are saying B right here. Okay, so yeah, and technically I should probably say like right before meiosis 2 or meiosis 1 because it does happen before meiosis 2, just like way before, right? But the point is that it doesn't happen again. It happens before meiosis 1 to get our double set of DNA, and then we go down to 2 cells, but it doesn't happen again before we go down to 4 cells, and that's how we end up with the half amount of chromosomes in the end. Okay, any questions on that? Yeah,
So it does division twice, but not replication.
perfect. Yeah, exactly. Division twice, but not replication twice. Good. Okay. All right. So this idea of getting some genetic variation across all of the different daughter cells that come out of meiosis happens because of a couple of different things. And we'll talk about a couple of those in a very basic way here. So one of those things is called crossing over. Okay. And this occurs during prophase 1. Okay. We have those homologous chromosomes paired together right up next to each other. And what happens when sometimes when they get close to each other, the ones that are really close to each other actually can just kind of exchange some genes with each other. Okay, so one gene goes to the other gene comes to the other one. Okay, and so if it happened to be dominant on the other side, recessive on the other side, those can flip-flop places on the chromosomes, and that's kind of what this tangling is. Okay, so evenly exchanged pieces of DNA, but then those chromosomes aren't exactly the same necessarily, so they could kind of be switching out some dominant versus recessive genes or many other things too. So then those get pulled apart in anaphase 1. And so then some of the one that was on the one chromosome is going to stay there, whereas the other one is going to go the other direction. Okay, so the end result of this, the end of meiosis 2, is that each gamete contains one allele for each trait, which is what it should. But due to crossing over, it's really a mix of the alleles, and it's not going to match the initial chromosomes that started way up in the beginning of meiosis 1. Okay, so how does all of this relate to what we talked about in the simple kind of Mendelian genetics? Okay, one of those principles was the principle of segregation. Okay, so during fertilization, each parent passes on just one of the alleles for each trait. Okay, and so the product of meiosis 2 again is these 4 haploid cells, and they each only have one allele per trait. Okay, whereas those somatic cells have 2 alleles per trait. Right? And this is why we only pass on one allele, is because each of those gametes only has one allele for each trait, because it only has that one full set of chromosomes, not the pairs. It doesn't have 23 pairs of chromosomes, it just has 23 chromosomes. Okay, so that's kind of where this principle of segregation is being shown here. Principle of independent assortment. Segregation of each set of alleles is done independently from the other alleles. Okay, meaning that if a couple of alleles were next to each other in grandpa's genes, okay, then it doesn't mean that those alleles are going to stay next to each other when they get down to the offspring or to the gametes, because they could have flip-flopped a little bit during that crossing over process, right? So just because we started with alleles together on the same chromosome or chromatid doesn't mean they're going to stay together. Because of that crossing over process, we could do a little bit of flip-flopping before they get divided into those 4 gametes. Okay, so when that crossing over again is happening during prophase 1. Okay, so to get into that crossing over thing a little bit more, we'll kind of walk through this slide, and then we'll watch a short video on it here. So, okay, All right, so if we look at these chromosomes up here, okay, so in number one, DNA replication has already occurred. So we have 2 full sets of DNA. Okay, so there's 2 of the green ones, 2 of the purple ones. Okay, so we got 1A and 1B. Okay, as we move down, we go to that prophase 1 where they're right up next to each other. We see some of this mixing occurring, this tangling. Okay, that's what that crossing over is. All right, so the original grouping of alleles gets all mixed up because of crossing over. Okay, and so then when we finally move down to here, when those chromosomes start to pull apart from each other, now we have 3A doesn't look like 1A anymore, right? It's different because it's got this part of the green one and this part of the purple one went to that one, right? So that's really what the crossing over process is. So then when all 4 of these are split into their own cell at the end of meiosis 2, now we really have 4 different chromosomes than what we started with. In this case, in this illustration, these ones are still the same, okay? But these 2 in the middle are different than what we started with. Okay, let me show you just a brief little video that kind of walks us through this. Okay, so yeah, so then in the end, what we were really talking about is kind of back to what we were talking about for the last exam, which was protein synthesis, right? Because all of these genes, what they're really doing is dictating how we make proteins, right? And which amino acids we call for, how those proteins come together. And that's really what gives us all these different traits that we're talking about with dominant versus recessive and all of those things, right? So just as kind of a reminder, right, that what we're talking about in chromosomes is these DNA. That DNA is going to call for certain RNA. That RNA is going to call for certain amino acids, which are then going to come together to make certain proteins, right? And again, it's those 3 DNA bases going to lead to 3 RNA bases, and then that's going to give us 1 amino acid using that table. Okay, so what happens when that DNA gets changed, right? And that's really what a mutation is. A genetic mutation is when that DNA gets changed, okay? And so there's a couple different types. If we have what's called a germinal mutation, this is a mutation that actually happens in an egg or a sperm cell or one of the cells above that, those germline cells that are going to eventually make the egg or the sperm cell, okay? And if that's the case, that we can pass those on, right? We can pass that genetic mutation on because that's the type of cell that actually becomes our offspring, okay? If it's a somatic mutation in a body cell, we can't pass those on, but it could definitely still cause problems like tumors and just malfunctioning structures and things like that. Okay, lots of reasons for DNA to become mutated. Definitely mistakes in DNA replication. Okay, that process is incredibly happens incredibly fast. Is very detailed, right? And most of the time there are mistakes made, but there's certain enzymes that are actually looking for those mistakes, correcting them as they go. Okay, so most of the time that isn't the case, but it definitely can happen. Also, certain exposure to different things that can damage DNA. So UV light, radiation, certain chemicals, all of those different things can potentially damage our DNA. Okay, so the different types of mutations that we can have. The first one's called a silent mutation. Okay, so silent mutation is we change the DNA structure in some way, right? Maybe a C becomes an A, okay? But in that case, it's one that actually ends up definitely changing the RNA. But as we look back on this table here, we can see that there's a bunch of RNAs that call for the same amino acid, right? So in the end, it doesn't really change the protein, right? Because the protein, the amino acid stays the same. There's no actual change in the protein, okay? So that's called a silent mutation because the DNA changed, but it didn't actually change the protein at all because we ended up with the same amino acid. Okay, so example here is CGC to CGA. Both of those are in the grouping that's going to give us arginine as the amino acid. And so it doesn't change. Okay, so that's kind of a freebie mutation. Okay, but then there's what's called a missense mutation, which the DNA changes. Okay, it does result in a different RNA, and it also results in a different amino acid, which changes the structure of the protein. Okay, so for example, the one we had talked about, sickle cell anemia a little bit. Sickle cell anemia is rare in that it's just a single amino acid different. Okay, other mutations are multiple amino acids, but this is one where it's just a single amino acid that changes. Okay, so the DNA gets changed from the CTC to CAC. So that gives us a different RNA, gives us Yeah, and then gives us a different amino acid, goes from glutamine to valine. And that's that right there, just that one amino acid is what causes this disease. There's not a ton of diseases that are that simple, but that's one example of one that is. Okay, so we got silent mutation, missense mutation. The next one, I'll give you a second to finish writing that down. I see some of you still writing. Okay. All right. The next type is called a nonsense mutation. Okay, in this case, again, the DNA changes, which changes the RNA. And instead of getting an amino acid, we get a stop codon instead. Okay, and then what ends up happening there is the protein just doesn't get formed. Okay, so we just have a missing protein. Okay, which is the case with certain things like brown eyes versus blue eyes, with hemophilia. Both of those are examples of things where we just don't make that protein that we're supposed to make. And so obviously the brown eyes, blue eyes one doesn't hurt us, but hemophilia obviously does, because we have certain clotting proteins that just aren't in our blood anymore. Yeah. Go ahead. Say that again. Yeah, blue eyes are a mutation. Yeah. So mutations aren't always bad, even though they sounds like a bad word. It's not always bad. Yeah. Okay. All right. Other questions on that? Okay. So when we look at the full set of chromosomes, well, the term for that is a karyotype. And a karyotype, again, is this complete set of chromosomes, 23 pairs, so 46 total. Okay. Each chromosome stains slightly different. Okay. And so when you put it into a petri dish, put different types of stain in there, they look a little bit different, and they're different sizes as well. Okay. And so they can each be identified. Okay, and so of the, so there's 22 pairs total of what are called autosomes, and then there's the 1 pair total of sex chromosomes. Okay, so that gives you the 23. And most of you guys know this, females have the XX for the sex chromosomes, whereas males have the XY. Okay, the Y chromosome is what gives a lot of the male dominant features. Because one is only present, and yeah. And so let's do one more Punnett square example here, but using these sex chromosomes instead. Okay, and so it's a little bit different because your offspring you're writing in are some are going to be male, some are going to be female, and so let's look at this one. So this is the hemophilia example. Okay, so normal clotting is the capital F on the X chromosome. Okay. Hemophilia type A is the lowercase f on the X chromosome. Okay, so here's kind of a what it looks like, how you translate genotype to phenotype. Okay, you can refer back to that after you fill out the Punnett square in the next slide. Okay, so the mom's genetic makeup is like that, genotype's like that, whereas the dad's genotype is like this. Okay, so you only need one of the, so you guys can work that out through the phenotype, but I'll let you go ahead and fill out that Punnett square with the people on your team, and then go ahead and answer this question again on your clicker, the likelihood of having a hemophiliac male. Okay, so whenever you're ready, talk through that with people on your team after you've worked that out before you send in an answer, but then send in that numeric answer on your clicker. So the percentage, that percentage number again? Yeah, just like we did before.
Am I a genius?
All right, go ahead and send that in if you haven't yet. All right, got 53, couple more, send it in. All right, you can just hit send if you haven't quite got there. 61. All right, about 4 more of you still. Make sure to hit the send button. Okay. All right, so the majority of you, 94% of you are saying 25%. Okay, so this is how you would fill that out. We've got the genotypes down there. You've got to translate those genotypes over to phenotype. Okay, so normal female, 2 out of 4. Hemophiliac female, 0 out of 4. Normal male, 1 out of 4. Hemophiliac male, 1 out of 4. Okay, in this case, it's not possible to have a female with hemophilia, right? The only way it would be possible is if the dad also had hemophilia, right? Because he's going to pass on that X, and that X needs to have the lowercase on it. And for the female to have it, they both have to be lowercase f, right? So one has to come from dad, and so if he only has one to give, if it's the one with the capital, then there's no way that you're going to get a hemophilia in that case. Okay, same thing with color blindness. We'll talk more about that one later. But yeah, we're pretty much out of time, so we'll finish up these last slides next class. See you guys on Wednesday. Have a good day. All right, see you later. Bye. See ya.
I got it from a friend that is.